We've looked at messenger RNA and the processing of messenger RNA. If you didn't see that, you should go back and look at the messenger RNA lecture. Now what we're going to focus on are these other two types, the transfer and ribosomal RNAs. Not going to as much processing that we're going to get into with them, and once we get into the ribosomal RNA, it's going to lead right into the introduction of translation. So the two topics are going to pretty much be joined together, although we'll keep translation as a separate lecture. So right now we're focusing on these two types, transfer and ribosomal. So again, coming back to here, genes code for RNA. They don't just code for messenger RNA, they code for messenger and transfer and ribosomal. The messenger RNA contains the code for the amino acids, but the amino acids need to somehow be involved in this process, and the transfer RNA is going to be doing that. And then all the amino acids are going to have to be joined together. The ribosomal RNA is going to be working on doing that. So we still have RNA here, and so we just have RNA with a 3' and a 5' end, like this. So now we're going to look at processing of transfer RNA. The job of transfer RNA is going to be to carry with it, in the cytoplasm of the cell, single amino acids. So it carries single amino acids. The transfer RNA is also going to have, as part of its own structure, so we say it carries this, but it has an anticodon. I'll say it possesses an anticodon. And we'll get in a second, we'll explain that and relate it to the messenger RNA. The transfer RNA as a characteristic is going to have 3 loops, and we're going to get into this in a second too, 3 loops and 4 stems. All right. So what's going to happen with the transfer RNA in terms of processing? Well, the transfer RNA starts off as a short piece of straight RNA, like this, single-stranded RNA. And then what's going to happen is that this single-stranded piece of RNA is going to be folded. So let's say, for example, here's still our 5' end. Here's still our 3' end. And what's going to happen here is that part of it is going to hydrogen bond with itself. Okay, so there's going to be hydrogen bonds that will actually join one part of the strand to another part of the strand. Okay, that region there, where the hydrogen bonds are, that's called a stem, a straight region with hydrogen bonds. This region down here, there are not going to be any hydrogen bonds in there. And that region is going to be referred to as a loop. So I have a stem and a loop. So you can see this is how it's just kind of folded, but not completely joining together. That part doesn't. The overall structure of a transfer RNA, we said, is going to have now 4 stems and 3 loops. And so one simplistic way, this isn't the actual structure, but this is called a cloverleaf diagram, a structure of how the RNA in general looks. What we have is hydrogen bonds here, 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 and here. All right, these are the 4 stems. And then these areas here are the loops. And the 2 ends end up together on this top stem. Okay, that stem, that top stem or acceptor stem, is where the amino acid is going to be attached. So the first thing that happens to a transfer RNA is that it's folded into these stems and loops. The second thing that's going to happen is the amino acid is going to be attached to this 3' end here. So we're going to have some specific amino acid attached right there. It's going to carry a single amino acid. That's going to happen after the fact. So after it's folded, then the amino acid will be attached. There is a whole process through which that occurs with an enzyme called the amino acyl transfer RNA synthetase enzyme. It's an enzyme that's going to bind to a transfer RNA. It's going to bind to an amino acid. What it does first is that it binds to ATP, activates the amino acid. It adds energy, essentially, to the amino acid. And then it can attach that and form a bond between the two. It has to be sure that this transfer RNA is the correct transfer RNA to carry a specific amino acid. So if you recall, there are 20 different amino acids. What we're going to have as we get along a little bit further, we're going to look at the codes and the codons, is we're going to find that there are 4 nucleotides. In this case, the A, U, G, and C. A codon is going to be made up of 3 nucleotides. So we can organize these letters, A, U, G, C, into groups of 3. And there are 64 possible codes. Now, 61 of those are actually going to code for amino acids. 3 of them are called stop codons. Now, those codes, let's say down here, I'm going to pull this one out actually specifically. So let's say that's one of the codons. Okay, that's the code in, let's say this is our messenger RNA down here. That's coding for a specific amino acid. This particular codon here, this one that I highlighted here, the code AUG, and that's in messenger RNA, is going to code for the amino acid methionine. Now, an amino acid is going to be carrying, a transfer RNA can be carrying one of 20 different amino acids. One of those would be methionine. Now, methionine then has that transfer RNA with that methionine has to match up with this codon. So you have to ask yourself, how can they kind of pair up properly? Well, if you think about complementary strands, say in DNA, an A binds with a T, a G binds with the C. That's complementary base pairing, and that's what's going to happen here as well. So along the transfer RNA, we're also going to have something referred to as an anticodon. And so this bottom loop here is referred to as the anticodon loop. Now it's all nucleotides. It's not just 3 nucleotides here, but we are concerned with 3 nucleotides. These are the 3 nucleotides that pair up with the 3 nucleotides in the messenger RNA. So we actually have transfer RNA binding with the messenger RNA through these particular nucleotides. And so I'm just going to do a quicker little simple drawing of it here to give us the idea. So if we had methionine up here, we can have sticking off the anticodon loop a U, an A, and a C, right? So U and A and U, C and G, that's a correct pairing, right? This is the purpose of the anticodon loop. The anticodon loop, which has the sequence, does say 2 things. One is that it helps this enzyme, which you're not going to be responsible for in my class to necessarily know all the details of it. But it's called this aminoacyl transfer RNA synthetase enzyme that's going to bind here, read the anticodon loop, potentially in certain cases. It can also read these loops. There's one called a D loop and a T-Cy-C loop, which again, we're not, on our course, we're not getting into. It's a slightly more advanced cell biology topic. But essentially, that enzyme will read information from all these areas. And it will know that there's only one specific amino acid that can be attached to it, and it will attach then that specific amino acid. If it makes an error, there's no way down here of knowing if this was lysine. For example, if it said UAC, then lysine would go in where it's not supposed to, where it's supposed to be methionine. But in this case, hopefully that process works properly, and it's always the correct amino acid being put in the correct place in the sequence of amino acids of the new polypeptide. All right. So a transfer RNA is modified by folding it into stems and loops and attaching an amino acid to it. one of the important parts of the transfer RNA is this anticodon loop, the bottom loop. And its job is to pair up with a codon in the messenger RNA. So here we have, the bottom here is messenger RNA. This is our transfer RNA, this guy here, right? It's got its stems, and then our 3 loops. And that bottom loop will eventually pair up. It's not going to do it all by itself. That's where the ribosomal RNA will come in, but that is the next part of this for the transfer RNA. So what we want to do is, start to bring in the ribosomal RNA as kind of the last part of this, and then we can, begin the translation process. In order to talk about ribosomal RNA and its modification, in some ways it's very complex, but in other ways, in terms of how we're going to discuss it, it's going to be even a little more simple, potentially, than some of these other types. And that's partly because the structure of the ribosomal RNA is so complex that we really can't go into the detailed type structure like I have here. So what we're going to do now is look at the rRNA, or ribosomal RNA, and its processing. And then we'll kind of end with this section of the material, and then move on eventually to translation. So what we'll have is a pre-ribosomal RNA, pre-rRNA. That ribosomal RNA will then typically be cut into pieces, right? And the pieces will be different sizes in terms of length. Okay, so the pre-ribosomal RNA will be cut up, and it'll be cut up into pieces that are 28, 18, 5 and 5.8, 5.8, and then just 5, okay, like this. So this S is a unit of size, of measure. It's a Svedberg unit. This has to do with the overall density of the pieces, which has to do with their size. Okay, so 28 is the biggest, 5 is the smallest, and the pre-messenger, or the pre-ribosomal RNA, is then cut up into sections of these particular lengths. Different genes might code for different types of the ribosomal RNA, again, and that's a slightly more advanced topic of which genes code for. Some of the multiple ones are actually end to end to end to end, and they're just cut out in sequence. Others are coded for by different genes. But we have some genes that code just for this ribosomal RNA. Now, what's going to happen is the 18S ribosomal RNA, this particular one, is going to stay by itself. But the other 3 types, they are going to join together. So the 28, the 5.8, and 5S ribosomal RNAs, they're going to join together. They're going to join together with proteins and form something called the large ribosomal subunit. And that's what we're going to kind of get into more with the structure of a little bit. And then the 18S will join with proteins, and it will become what's called the small ribosomal subunit. Okay, so we're going to have 2 subunits of ribosomal RNA, large and small. The small will be the 18S alone, and then the large will be the other 3 types all combined with one another. What we're going to do, though, is right now focus on just the large subunit, and it will lead us then into the translation process. So, the large subunit, the one with the 28, 5.8, and 5S, combines with proteins and forms this structure that has 3 binding sites. These 3 binding sites are referred to as the A site, the P site, and the E site. So these are binding sites along the large subunit. Each of these binding sites are unique. They are going to bind transfer RNAs at a particular type of transfer RNA at a particular location. So ultimately, what will happen is, like I said, this transfer RNA isn't going to pair up with the messenger RNA all by itself. It has to associate with a small ribosomal subunit and this large subunit together. So the large one is separate from the small one at one point in time. Then they assemble along the messenger RNA, and they form a really large structure here that we call our ribosome. The ribosome is a cellular organelle, and it is the one cellular organelle that isn't made up of membranes. And we see ribosomes in eukaryotic and prokaryotic cells, and the overall structure is very similar, slightly different subunit, the length here, but overall they're pretty much the same. They do all the same things. Everything functions, and the rules are pretty much all the same for everything among different types of cells of different organisms. The parts here and what they actually bind. So to the A site, we're going to get into this. To the A site is going to be a transfer RNA is going to bind here with a single amino acid. So A, kind of for just amino acid, one amino acid. In the P site, you can have a transfer RNA bound to this site. But to the P site, we have a peptide or polypeptide. So we have multiple amino acids all joined together with peptide bonds. So P for peptide, and this is what we're going to see happen in binding to this site typically. And in the E site, you can think of it in a couple different ways. In the E site, what we would find there is an empty transfer RNA. There's nothing attached to that transfer RNA. There's no amino acid. There's no peptide. Now, there are never 3 transfer RNAs attached to this large subunit all at the same time. There's only 2 at a time. So it could be the P site and the E site, and there's nothing here in the A site, or it could be A and the P, and there's nothing in the E site. It typically works like that. Right? There's usually then something always in the P site. But as it reads a new codon, a new transfer RNA comes in with a single amino acid, this empty one gets kicked out. Then a peptide bond is going to form between these. That's going to be the translation process. And then the whole thing will shift over. One of these will then lose its polypeptide and be empty. And then this site will just remain open. And then the whole process is going to continue. It's this active process that's going to be working its way down the messenger RNA, reading codon by codon by codon by codon. New codons in this A site are going to be read because messenger RNA is going to end up coming through here. So what we're going to do is now, ultimately in this last part, is we're going to put together all the different types of RNA and then get into the protein synthesis process, the process that we refer to as translation. But to wrap up the ribosomal RNA part, what do you need to know about ribosomal RNA? Like I said, we're not getting into structure of it. The structure is fairly complex. There's a lot of stems and loops, and it's very unique, and there's all these different types. So we're not going to really go into that, and you're not going to really be trying to draw that or anything. You should be able to draw or sketch a simple piece of messenger RNA. It's just a line with A's and U's and C's and G's and so forth. You have a transfer RNA. You should be able to sketch that. You could just draw these little loops and stems and label them as stem or loop. That's pretty easy. The ribosomal RNA we can't really draw, so we're just doing the subunits here. Make sure you could just draw, say, a circle, and you're labeling it 28, 5.8, and 5. That's the large subunit, and the 18S in eukaryotic cells goes with the small. That's 16 in bacteria or prokaryotic cells. But that's kind of what we have. And then we have these 3 binding sites in the large subunit, the A, the P, and the E site. And we're pretty much set up then at this point, right? So as far as other types of processing for ribosomal RNA, we're pretty much just saying it's folded, it associates with proteins, it makes these subunits, and we're not going into any further detail. Right, so hopefully you got that down. If not, you go back and review some of it. Look at the messenger RNA part, the transfer RNA part, if you're not sure of them yet, because now we're going to get into translation, and it's important that you are familiar with each of the different types of RNA before we start really putting them together and going through the steps of that process. That's it.